Hello and welcome. In this video, we are going to talk about answer writing with a specific reference to botany and zoology. Both these subjects are sister disciplines within the ambit of life sciences, and the principles of effective answer writing apply identically to both the subjects. In this video, we are going to grasp those 12 principles of good answer writing in these subjects, and then I'm going to explain in detail with example each principle, what are the do's, what are the don'ts. The whole purpose of this exercise is to help you achieve your fullest potential in writing answers and performing credibly for the civil service examination. I would like to start with this quote from one of the most influential management thinkers and a renowned author, Jim Collins, who writes in his book, Good to Great, good is the enemy of great. What he basically means here is that when you settle to be just good enough, you are unable to fulfill the fullest potential that you have. You are unable to meet your performance matching your potential. You always remain a little behind your potential. So when you are wanting to achieve the fullest potential in terms of your performance, you don't have to settle for good enough. You have to strive for a great performance. So what are those 12 elements of a great answer in the context of botany and zoology optional? First, I'm going to read out and then I'm going to explain in detail with proper example. So first thing is staying focused. Each question has a couple of query areas. We have to underline those query areas and we have to stay entirely around those query areas throughout the answer. Second, paying minute attention to the wordings of the question so that we exactly know what we have to write on the query area. Is it analyze? Is it describe? Is it comment? Is it compare? Whatever it is, we need to know very clearly, and that is how we are going to treat the query area. Third is staying clear. Whatever you write with simplicity, write in short sentences and inform the examiner with clarity. Next, it is a science subject. You ought to be correct all the time. Inaccurate answer kills the impression and fetches 0 marks. Fifth is you have to be specific and you have to substantiate your observations. That essentially means that you do not have to make a statement that you cannot substantiate with example or facts. Similarly, whatever you are writing, write in specific terms rather than vague terms. You have to be concise because since 2010, all the questions are being asked in the short note format. You have very limited time and very limited space to write your answer. So concise answer is the only way to finish your paper. Next, you have to give complete information. By this, at a simple level, I mean, if you are writing about glycolysis, you have to write about all the 10 reactions. If you are writing about Krebs cycle, you have to write all the 8 reactions. That is the meaning of being complete in terms of information. Then you have to use the language of the subject. Krebs cycle is Krebs cycle. Citric acid pathway is another name. Tricarboxylic acid pathway is another name. But these are the names that you can give to this cycle. That is the language of the subject. I'm going to illustrate it with certain examples a little later, but let's see what are the other points. The next point is you have to structure your answer well. That means divide your answer into introduction, body, and conclusion. Also, depending on how many query areas are there in the question, 2 query areas, 1 query area, 3 query areas, accordingly, within the answer, you have to create headings. In science subjects, you can very well create headings. I do not know whether it will work in humanity subjects or not. I do not claim to be an expert of humanity subject, any humanity subject for that matter. So there, whatever the teacher has to suggest, you follow. But I'm presuming that if you are watching this video, you are an aspirant with botany or zoology optional, and here heading and subheading are pretty much expected from you. Not only it is tolerated or accepted, but it is actually actively expected from you, and it is a welcome addition to your answer. Lastly, you have to learn the art of giving proportionate weightage to different parts of the question. all the parts of the question do not always need to be given equal weightage. Next, you have to illustrate with examples, charts, and figures. 11th is you have to make it easier for the examiner. So you use headings and subheadings. As I told earlier, it will make it easier for the examiner to navigate through your answer. You have to keep the drawing and description close to each other, and you have to underline the keywords. The 12th point is packing a punch. Packing a punch basically means trying to do something that your even most capable competitor is unlikely to do. You do not know whether your capable competitor is going to do or not, but at least you should be doing it, and you should be able to identify what my even capable competitor is less likely to do. It can be anything. For example, it can be some originality when it comes to write an introduction or write a conclusion, giving attention to certain details which you think most of your competitors are likely to ignore. You may apply a little extra depth, but not excessive, because you can anyway not accommodate it within the format of a short note. You can here and there use updated content, and I'm going to give you example of every single thing. So stay till the end of the video so that I can communicate to you in the fullest detail all that I want to share on answer writing. You can be application oriented, especially in the concluding parts of your answer. You can draw better drawing. And my bonus point, the 13th point, is you have to be really fast, because for one short note you are going to get 10 minutes or less. So while doing all of these things, you also have to maintain speed. I'm going to talk about the speed a little later, what causes a lot of people lose speed in the answer writing and all that. I'm going to discuss all of that. Let's start with the very first point, that is staying focused. Let's read this question that was asked in the year 2017. The question was, what is Hardy-Weinberg law? How this law explains the mechanism of evolution. So there are two query areas which I have highlighted in yellow. One, explain what is Hardy-Weinberg equilibrium state assumption and what is the law that dictates this equilibrium. Second part is how this law explains the mechanism of evolution. So there are going to be two parts, and then you do not have to treat absolutely anything else. Do not go into Darwinism, do not go into new synthesis of evolution. Although Hardy-Weinberg equilibrium is very much a part of the new synthesis of evolution, but you do not even have to go in there. Just stick to the core demands. That is what staying focused is. Why it is necessary to stay focused on the core demands of the question? Because every single answer that you are going to write is a short note. Now, in a short note, you have no word, no sentence to waste. So every sentence, everything that you write has to cater to the core demand of the question. Hence, stay focused. That's the first step. And this is the example. Second, pay minute attention to the wording of the question. For example, in 2019, this question was asked. Discuss the role of molecular techniques in the study of contemporary systematics. So this is a question that was asked in evolution and systematics. In systematics, there is a topic, molecular taxonomy. That is where this question has come from. But the topic is molecular taxonomy. And the question uses the word contemporary systematics. So you have to define here what is systematics, and within the overall scheme of systematics, where does taxonomy fit in, and what is contemporary systematics? What is the approach of the contemporary systematics? So your syllabus does not use the word contemporary systematics. Your syllabus uses the word molecular taxonomy. But you have to focus upon contemporary systematics. Of course, what you have learnt in molecular systematics will be forming the bulk of the answer, but the answer has to open with systematics, and what is contemporary systematics. So that ways you have to pay minute attention to the wordings of the question and then, of course, discuss. That means what are the roles which molecular technique are playing in the field of systematics. In simpler words, what is the role of molecular taxonomy in contemporary systematics? Accordingly, you have to word your answer and craft your answer. Third is very interesting point, that is be clear. Take two statements which are side by side. One is a questionable. I'm not saying it is wrong, but I'm questioning it for its efficacy. It is good enough. The right one, I would say, is very good or a great one. So the first one is transcription is the first of several steps of DNA-based gene expression in which a particular segment of DNA is copied into RNA by the enzyme RNA polymerase. Both DNA and RNA are nucleic acid which use base pair of nucleotides as complementary language. This is something that has been taken from internet. But you have to understand this is the pitfall of relying solely on internet. So what will be a good and clear way to express the same information? Simple. Transcription is DNA template driven RNA synthesis by the enzyme RNA polymerase. That's all. It is the universal early stage of gene expression in all cells because all cells have DNA as the genetic material. So gene expression will always be having transcription. Whether the gene product is an effector RNA or the gene product is a protein, transcription is bound to take place. So when you are writing, write clearly. Do not write vaguely. So this I would not like. The other one is just an example how you can write with clarity. I hope I have been able to illustrate this point. Next one is always and always be correct. In sciences, this is one of the most fundamental things. Let us check two statements. Mitochondria is called the powerhouse of the cell. What's wrong here? First, mitochondria is a plural expression. Therefore, you have to use are. Mitochondria are called the powerhouse of the cell. Second, because it is the site of most of cellular energy generation. Remember, energy is neither generated nor destroyed. It is ATP that is generated in the mitochondria. Energy is already there. That energy is used to generate ATP. So how we have written? Because most of the cellular ATP is synthesized here. The next one is being specific and being able to substantiate. Whatever you write, you have to write specifically. So I'm going back to the same question which I had discussed in the beginning part of the discussion. What is Hardy-Weinberg law? Discuss how this law explains the mechanism of evolution. Two ways. The Hardy-Weinberg law explains the evolutionary process at the level of a population and a statistical model of organic evolution. That is a vague way, that is a non-specific way of writing about how does Hardy-Weinberg law help us understand the evolutionary changes. See a more specific answer, more specific statement. It is not the entire answer. The Hardy-Weinberg law identifies 5 fundamental factors which change gene frequencies. So what do these factors do? They change gene frequencies and cause populations to evolve. And simply name those 5 factors as Hardy-Weinberg had given. Mutations, migration, natural selection, non-random mating, and genetic drift. So always write specifically, no vagueness, and substantiate. If Hardy-Weinberg explained 5 factors, name all the 5 factors. That is being specific and substantiating. Moving further, be concise. That basically means give more information per 10 words. Most of your answers are going to be 120 words, 150 words, at a max 180 words. So per 10 words, you have to give more information than your competitors. Read the 2 parts. questionable part first. The electron transport chain is a series of complexes that transfer electrons from electron donor to electron acceptor via redox reactions and couple this electron transfer with transfer of protons across the membrane. The electron transport chain is built up of peptides, enzymes, and other molecules. Again, vague. You have to be concise. You have to distill to most relevant information, and it is as simple as this. The electron transport chain has 4 complexes and 2 mobile electron carriers. That's it. Name the 4 complexes: NADHQ oxidoreductase, succinate dehydrogenase, Q cytochrome C oxidoreductase, and cytochrome C oxidase. Then name the mobile electron carrier. Mobile carriers are quinone and cytochrome C. That's it. So write concisely, and as earlier discussed, write with a specific information. Next, give complete information. Same example. Here it says the electron transport chain is built up of peptides, enzymes, other molecules. No, not like this. Simply, it has 4 complexes and 2 mobile electron carriers. That is complete information. There is nothing else that you have to add here. There are 4 complexes, hence only 4 complexes has to be mentioned. So give complete information. As I told you earlier, let's say that if Krebs cycle has got 8 steps, mention all the steps, the substrates, the product, the nature of the reaction, the enzyme, NADH or FADH2 produced or not. That's it. So always give complete information. Next one is use the language of the subject. Let us see. Both these para explain linkage. So genes that are sufficiently close together on a chromosome will tend to stick together, and the versions, that is alleles of those genes that are together on the chromosome, will tend to be inherited as a pair more often than not. This phenomenon is called genetic linkage. This is a classic way of explaining scientific information in layperson's term. In optional subject, you have to speak the language of the subject. The same does not apply when you are in general studies, but in optional subject, you have to stick to the vocabulary of the subject itself. So here we are. Genetic linkage refers to 2 or more than 2 genes failing to show Mendelian independent assortment. So Mendelian independent assortment is language of genetics. So failing to show Mendelian independent assortment ratios and inherit largely in parental allelic combinations. So parental allelic combination is another word because they are located on the same chromosome. Then you have to write such genes are called linked or syntenic genes. Syn means together, ten means thread. So together on a thread, that means together on a chromosome. So the things which are written in yellow are the language of the subject which must find a place in your answer. That is what is using the language of the subject. Moving to the next principle, structure it well. First and foremost, write a short introduction, then build the body, then write a proper conclusion. Divide it in headings and subheadings. In subjects like botany and zoology, headings and subheadings are pretty much appreciated by the examiner because it makes it easy for the examiner to navigate through your answer. And then learn the art of giving proportionate weightage. I'm going to illustrate my point on proportionate weightage. When I say proportionate weightage, I do not mean equal or unequal. It depends on the question. Let's see 2 questions and you will get it. One question is, give the structural formula of an amino acid and show how peptide bond formation occurs. Also explain how amino acids differ from each other. So first and foremost, you will talk about the structure of amino acid. So even though it is saying that draw only the structural formula, explain what is an amino acid made up of. So this is one part of your answer. Second, show how peptide bond forms. So that is the second part. So you will have to show that condensation reaction where two amino acids react together and a peptide bond is formed. And third, what are the parameters based on which amino acids differ from each other. So these are three parts, and these three parts can get more or less equal weightage. But will the same apply to this question? This is a 2014 question. What are nucleic acids? Describe the structure and functions of various types of RNA. Now it is obvious that what are nucleic acid can be concluded in 3 to 4 sentences. And if you include DNA, RNA, 5 sentences. But where the rest of the answer is going to be? The rest of the answer is going to be on a structure and function of various types of RNA. So this is a classic case of non-equal weightage, that the first part gets less weightage, second part gets more weightage. If I give you another example, so let us talk about Krebs cycle and its regulation. If this question comes, the core of the Krebs cycle is going to get greater weightage and its regulation is going to get a lesser weightage. So learn the art of assigning proportionate weightage, and this art comes when you have got solid concept in the subject. Moving to the next principle, illustrate with examples, charts and figures. So where to use example? Whenever you are talking about a phenomenon, example is a must. Whenever you are talking about a process, an organization or diversity, charts are a must. Right? So what are the types of cells present in blood, or what is the composition of blood? You can make a chart. If you are explaining the process of, say, ozone depletion, a chart can be made. If you are explaining a physiological process, a flow chart can be made. Cell cycle regulation, a flow chart can be made. Why? That's a process. Then if you are describing a structure, mouth part of insect, larval forms of crustacea, larval form of echinodermata, these are all a structure, and there you have to draw figure. So structures are illustrated with figures, phenomena are illustrated with example, and processes, organizations and diversity are illustrated with help of charts. So illustration is needed in biology because biology is an illustrative science. You have to substantiate, hence the need for illustration. Next, make it easy for the examiner. So this is a very interesting, and it is actually a humorous formula that I have written. Let me explain. S stands here for a score. So I'm saying that a score is proportionate to happiness of the examiner. So your score is going to be proportionate how happy the examiner is with you for that particular answer. So it is entirely up to you how easy you make it for the examiner. Use headings and subheadings, underline the keywords, and always keep the diagram and description close to each other so that in one flow the examiner gets to read the description as well as gets to see the diagram. So that is what it is going to be. Last one is packing a punch. So packing a punch is basically do anything relevant. So relevant is the key. In order to be different, you cannot go in the realm of being irrelevant and put anything. So a lot of students ask me, sir, the current that we get from the newspaper, can we use it in botany or zoology or no? Answer is mostly you won't get an opportunity to do that. But when you read from standard textbook, or when you are referring to good resources, you will have lot of relevant, updated, deep information which here and there in a selective manner you can use to get that attention of the examiner and establish that you are better prepared than most of your competitor. So if you can pack a punch here and there, that will be terrific to get that extra edge compared to your even most accomplished competitors. There are certain things that you can think to pack a punch. So a number of things that came to my mind. First, originality. So write something which you think is so original that your competitor is unlikely to use. So the greenhouse effect refers to the gradual warming of the Earth's atmosphere and hydrosphere due to radiative forcing. Radiative forcing comes from IPCC report. The latest IPCC report uses the word radiative forcing of infrared radiation. So when you read it in general studies, you can use it here under ecology and environment section. That is originality. Originality does not mean that you will coin a term on your own. You and I are nobody to coin our own terms. So refrain from that. That will be absolutely counterproductive. But when you get something authentic from a good source, and if you find an original way to use it, do use it. It will add a punch to your answer. Next, attention to details. So the entire world almost blindly accepts the fact that DNA replication is a semi-conservative process. But do you realize that it cannot be semi-conservative if we are talking about replication of a single-stranded DNA? So why not pay attention to such details and write something like this, that DNA replication is a semi-conservative process in all cellular forms, that is bacteria and eukaryotes, but in single-stranded DNA viruses such as 5X174, DNA replication is conservative. So this is paying attention to detail. When every single competitor of yours is going to write that DNA replication is a semi-conservative affair, you can add that extra bit of information to get that edge. It is possible, trust me, topic after topic, answer after answer. And I can actually illustrate had I been having more time in this video. Moving next, greater depth. When your entire bunch of competitors are going to write that in eukaryotes there are 3 types of RNA polymerases, polymerase 1, polymerase 2 and polymerase 3, why not add that in plants there are 2 more types of RNA polymerases which are related to RNA interference. So that is slight greater depth. So I'm not saying you have to go into the molecular 3-dimensional structure of RNA polymerase. That is irrelevant detailing. But when one sentence can give an indication about extra depth that you have developed, that is an easy way to establish before the examiner that your knowledge is deeper than your competitor. So do not chase depth in irrelevant direction. Chasing irrelevant depth is just now explained, that when you get into the job of studying the molecular architecture of RNA polymerase, that is unnecessary depth. But this is an easier depth to gain. Again, from great sources, you can get this thing. And also your eyes have to be magnet for such information, that okay, this information I can use for my advantage. And that is how you will be getting these information to be used in your answer. Next, updated content. By updated, I don't say that you have to read newspaper. Just read updated textbooks and that is all. So when you read updated textbooks, so you will be getting something like lysosomes are traditionally known as suicide bags of the cell, but they do not have primary role in apoptosis. That's it. So older textbooks will give you a full account of how lysosomes are responsible for cell death and all. But the fact of the matter is that lysosomes, since 1980s, it is known that they do not play a primary role in apoptosis. Any good international author book that has come in the market after 1980s clearly mentions that cell death is basically carried out by caspases, BCL family proteins, proteases, nucleases, but not lysosomes. This is updated content. So for updation, don't get into the journals, don't get into the newspaper magazines, just updated textbook or a material based on updated textbook. That's all. Next, application oriented. So give some application which is again unlikely to be used by your competitor. For example, DNA sequencing helps in making gene trees. Most of your competitors are not going to use this application of DNA sequencing. And then explain what is gene tree. So gene trees show evolutionary relationship of a single gene in either different species or members of a gene family. So this kind of application. Similarly, AFLP, amplified fragment length polymorphism, is an effective technique to use polymorphism to detect polymorphisms in DNA when no information about the genome is known. This is one of the most fundamental aspects of AFLP. But again, when I check the copies of test series, I do not see anyone clearly mentioning this application. So that clear application sense is again a sure shot method to add punch. Next is drawing better figures than your competitors. So you have to draw better figures than your competitors. It will happen only with practice, and a good figure is clean, proportionate, and it focuses on what is being asked, and it does not try to represent everything that a cell or a tissue or a part of the body has to offer. It simply focuses on what is intended to be shown there in that answer. So these are some of the ways. You can always think of more ways to add punch to your answer. So these were the 12 principles. It is with some diligence, some persistent practice, you will be able to develop that. Do not be impatient. Nothing builds overnight, but when you are practicing every day, these 12 pillars of good answer will very easily be a part of your normal answer writing. So that is what the pillars of good answer are, and I have tried my best to illustrate using examples. Lastly, that's my bonus point, be fast, because in 180 minutes you are going to write 17 to 18 short notes. So you are having only and only 10 minutes to write an answer. In 10 minutes and finite space given by the UPSC in its answer sheet, you can only write a limited amount. Now, most of the students tend to think we write slow. The problem is not writing slow, the problem is recalling slow. So you have to recall at a greater speed. And this recall at a greater speed requires that you have revised your subject matter for 4 or 5 times. How do you revise? When you have made self notes from very good sources. Very good sources can be our study material at Evolution, good textbooks which I'm anyway suggesting through my weekly series on syllabus discussion. So pick up a set of textbooks, make your self note. Pick up our study material, the latest one, make your self notes. But self notes have to be made because they are going to be concise enough which you can revise for 4 times, 5 times. And when you revise anything for 4 or 5 times, it will be helping you very speedy recall and thus very speedy answer writing. So one of the most crucial aspects of preparing for the main examination is to have a concise, fast revisable notes, which only you can prepare. No one can make that notes on your behalf. Good material can be brought at one place by good books, good material, good study material. But after that, the synoptical note is something that you have to do. And when you do that, the speed will not be a problem. So I hope you found this video helpful and it will get you on the right track on how to approach your answers. If it actually serves the purpose, I'll be very happy. Till we meet again, goodbye and good luck to all of you with your preparation. Stay safe, stay happy, keep working.